interfaces cohort this year. And so today I'll be talking about illuminating the mechanistic underpinnings of peroxisome biogenesis. And so just to make sure that we're all on the same page, peroxisomes are these organelles that are found ubiquitously throughout all eukaryotes. And so their primary functions within our cells are in lipid metabolism, so making different lipid precursors that will then go to the mitochondria. And then it's also in charge of lots of this oxidative stress regulation, particularly in the context of preserving mitochondrial health in our cells. And so, interestingly, as these organelles are becoming a little bit more studied, we're seeing how how they are more relevant in aging and neurodegenerative contexts. And so typically in cell biology, these peroxisomes are kind of thought about as these circular vesicular structures that have this little crystal core. But really, if you see these peroxisomes in like different electron microscopy like reconstructions for imaging, you can see that there are a lot more complex shapes here. And so there are there are lots of circular structures, but then there are also much more complex structures. And so traditionally, peroxisomes were believed to grow and divide, kind of like what you would think of like a mitochondria or chloroplast that maintains its cellular pool just by growing and dividing. And so you could see like a potential like growth and division event happening here where maybe this peroxisome might be pulling itself apart. But there's also this other type of way that peroxisomes are believed to like maintain their cellular pool, which is this idea of peroxisome biogenesis, and or de novo peroxisome biogenesis. And so what this would basically mean is that somehow the um, like a pre-peroxisomal vesicle would be able to bud from either the endoplasmic reticulum or a little bit more controversially from the mitochondria. And so um, the this process is characterized relatively well biochemically, where we know that there are these different proteins that might be involved in this, and we know that if this does happen, there's no contamination from ER membrane or ER luminal proteins that would be getting into the peroxisome at all. But we've never actually like proved this like through imaging. We've never like seen it kinetically happen where these proteins are in one compartment and then we're seeing them like actually move into like a small like vesicular structure that doesn't just have the peroxisome or proteins in it already. And so I'm basically with my project going to be looking to kind of understand how we might go from proteins that can localize to these other structures and then move into a mature peroxisome. And so the three proteins that I want to highlight quickly before I get into the rest of my talk are these PEX3, PEX16, and MCTP2 proteins. And so this PEX3 protein is responsible for inserting other peroxisome membrane proteins. The PEX16 is believed to recruit PEX3, so it would be a little bit upstream of PEX3 in this de novo peroxisome biogenesis events. And then there's this other protein called MCTP2, which is a little bit more newly believed to be involved in the membrane shaping mechanisms that would probably be required for budding. And so just to kind of like start or state the overall like goal that I really want to see is like to be like the first person to like definitively prove that these budding events are actually happening. And so to do this, my lab uses different microscopy methods over spatial scales. And so primarily that would include fluorescence microscopy, which it can be shown by this picture of the endoplasmic reticulum here, and then also of electron microscopy, which is the an EM slice. And so I like to think about these different types of microscopy as flying during the day versus flying at night. And so when you fly during the day, you can see lots of the native topology of the landscape that you're going through. But if unless you're like native to that spot or know a lot about that place, you don't really necessarily know exactly what you're looking at. Whereas when you're flying at night, you can label specific things and kind of see activity of where you are. but you might not know exactly where you are. And so the way that our lab is kind of bridging these two scales is through super resolution microscopy, where we can see super well-defined structures at much smaller scales. And with a picture of the ER like this, or the endoplasmic reticulum, when you bleach this, you can actually get down to single fluorophores. And what our lab specializes in is actually tracking these these proteins like through the ER membrane or just through the cell in general. And we can learn a lot about different types of proteins from their trajectories, characterizing their motion. And so in the first part of my talk, I'll be examining some of this dynamic organization, particularly with PEX16 and PEX and MCTP2, which we kind of know are already localizing to the ER. And so the first finding that I would like to present is that this MCTP2 protein localizes into stable subdomains in the ER membrane. And so what I mean by the subdomains is just that you can see that it's organizing into little clumps of individual proteins throughout some of these tubules and the other networking of the ER. And what I mean by stable is that basically, if this were to be a video, I'll play a video soon, but these proteins kind of just oscillate in place. They're not really moving much through the rest of the ER membrane. And so of a typical ER membrane protein, what you're seeing is something like PEX16 trajectories, where these proteins are able to move throughout the entire ER, but with MCTP2, for whatever reason, it just kind of stays in place. And so these splatter plots that I have here basically take the trajectory of one molecule and pin all of them to a shared origin. So you can kind of see where the proteins are moving. And so a protein that will move more will kind of, or that will diffuse more freely will kind of be going outwards, whereas something that's more confined, like MCTP2, will be stuck like this. And so additionally, in these areas where we can see there's MCTP2 puncta here, we can see that PEX16, which is the yellow here, displays more confined motion. And so specifically, we can quantify this and show that PEX16 has more confined motion when MCTP2 is overexpressed. And so these three graphs here show the mean square displacement of these different proteins. And so on the left here is the MCTP2, and you can see it's not really moving from one spot over time particularly, but then PEX16 without MCTP2 overexpression is kind of just moving very fastly. And then when you overexpress MCTP2 and track PEX16, you can see that there is still this very fast motion, but then there's also cases where it is not moving as fast, and then there are even cases where the PEX16 gets stuck. And so basically what we think this might be lending itself towards is that when these two proteins can co-localize, we can get a stable area that the ER can organize itself for peroxisome biogenesis. And so one more way to kind of quantify this is here we have these normalized fractions of the PEX16 trajectories in different diffusion states. And so basically here, what we can show is that when we overexpress this protein that is supposed to be the platform for peroxisome biogenesis, we're seeing a lot more slow diffusion of PEX16 in the ER membrane, whereas when we're just tracking PEX16 with normal levels of MCTP2, it's moving generally faster. And so the future work that I'm doing with this part of the system is I'm making these different CRISPR-Cas9 knock-in cell lines with PEX16 and MCTP2, so we can study the motion and co-localization of these proteins at the endogenous level to see how they're like natively expressed in cells. And then I included this second part because later this week I'm doing this fluorescence recovery after photobleaching assay, or FRAP. And so basically past work has shown that MCTP2 is able to recover and like diffuse back into the places where it is localized to. But obviously from what I've seen from analyzing its trajectories, if something is kind of oscillating around in place, won't really be able to diffuse into these new spots super quickly. So I'm interested to see how that turns out. And then so for the second part of my talk, I'll talk about a little bit, or I'll talk about this third protein, which is PEX3. And so PEX3 has been shown in various immunofluorescence and plasmid-based overexpression imaging experiments that it can localize both to the mitochondria and to the endoplasmic reticulum. And so I think there's the main question is like, which one of these or are both of these like real? And what is it doing at each of these places? And so to kind of show you what I mean by this differential PEX3 localization, I have three videos here that were all taken on the same imaging dish of cells that I transfected both with an ER marker and then a PEX3 overexpression marker. And so in this left one here, you can see both peroxisomes that could be seen in these bigger puncta with the PEX3 in red, but then also you see some individual PEX3 proteins. And so the way you can kind of distinguish them between that is based on like the blinking properties and the motion. But then in this other cell here, you can see that there are a lot more PEX3, there's PEX3 in peroxisomes localized both at the ER, but then also kind of free floating through the cytoplasm. You can see in some of these videos here. And then finally, in this last one, you can see PEX3 in some sort of tubular organelle. And so I don't have the specific mitochondria stained in this, so it could just be tubular peroxisomes, but I also have this other picture showing that PEX3 does seem to localize to like the distal mitochondria in some cells, which is super interesting. And so to further study this, I've made an endogenous PEX3 halo cell line, and so basically this is the same like approach that we would be doing with the PEX16 and MCTB2 endogenous lines, where I can study PEX3 at like the native expression levels within a cell. And so to do this, I'm using CRISPR-Cas9 homology-directed repair. And so this halo tag here that I have the structure of is basically just to replace like a fluorescent protein. And so with the halo tag, you can get like a generally like brighter, more photostable signal. And it gives you a like spectral flexibility with what cell permeable dye you want to use. So it's kind of just a little bit more multiplexed imaging that we're using it for. And so basically we would have a plasmid that has this halo tag, and then it has the overhangs that match the genome for this PEX3, and you can use CRISPR-Cas9 to knock it into the cell lines. And so here's like some initial result from that I have from this. And so in this endogenous PEX3 halo cell line, and so there's in this cell I overexpressed EGFP with a peroxisome targeting sequence SKL, which is in the green, and then the pink or the purple is the endogenously expressed PEX3 halo. And so you can see in most of these puncta, there's a pretty clear co-localization of the PEX3 and the SKL signal, which means that PEX3 is on peroxisome. So it's like, at the very least, that we know that is working, that's correct. But interestingly, there are some peroxisomes that you can detect that actually don't have any PEX3. So it's kind of interesting to be like, okay, how is this peroxisome like importing other proteins? And so I have my own theories about that, but more importantly, I guess for the specific talk here is that there are single PEX3 proteins that you don't see localized to a peroxisome. And when you actually look at the motion of these PEX3 molecules, you can see that they are stable in some places, and then they'll like kind of be shooting through tubules. And obviously, I don't have the ER labeled in this specific movie, but yeah, and so from the motion of this molecule, it's still not necessarily clear if it's in a like a vesicle or if it's in the ER, but this is like the first like ever proof that either that the pre-peroxisomal vesicles like with PEX3 like are real or that it could be in the ER. So whatever it does end up being is kind of a super cool result. But more interestingly, we can actually examine all of the trajectories that would be in this movie. And so in the purple here, I have the trajectories of PEX3 molecules where they are not localized to a peroxisome. And in the green, I have trajectories of PEX3 molecules that are on peroxisomes. And so we can look at the cumulative square displacements of these different PEX3 molecules. And so in the pink, I have, again, with the no co-localization with the SKL signal, and then the green means that the PEX3 is. And so with this cumulative square displacement is measuring is essentially the distance that the molecule is able to travel. And so you can see in general from taking the slopes of each of these individual lines that so the slope of the cumulative square displacement is the mean square displacement. And you can see by taking the slope of each individual trajectory that in general, when the PEX3 is not co-localized to the peroxisome, it is moving faster or diffusing faster, which, I mean, is a good sign because it's like obviously if it's in a peroxisome, it's going to be confined to a specific area. Whereas if it was moving through like the ER or the mitochondria, you can see it is will be moving faster. And I can quantify this similarly with the normalized fraction of PEX3 trajectories in these various diffusion states. And so you can see here that when it's on this left one, it's when it's by itself, not localized to the peroxisome, and you can see it's generally moving a little bit faster. With these, I will be doing like specific marking of the ER and the mitochondria soon. But I really wanted to like kind of parse together. I was most excited about doing it with the actual peroxisomes like labeled first to see if we can actually prove that there isn't that there is PEX3 not in these peroxisomes. And so the future work that I have coming up relatively soon with this is this new FRET assay that I'll be doing with the flow cytometer in our lab, basically to see during overexpression, like I can quantify the ratios of PEX3 localized to different compartments within the cells. So within peroxisomes, the cytoplasm, which hopefully it should be 0, and then the ER and the mitochondria. Eventually, once I get my PEX16 endogenous line working well, I'll be doing dual color tracking of PEX16 and PEX3 together to see if they are having any coordinated motion. That could be indicative of the formation of a budding subdomain. And then eventually after that, I'll be doing some DNA paint, which is basically just a super resolution microscopy method that allows you or to like count molecules in fixed cells. And so once I get an understanding of like how many proteins are actually ER localized through that with my endogenous cell lines, I can finally do the the correlative cryotomography to see if I can actually see a budding event for like the first time. And so I kind of just wanted to highlight some of the like multi-scale analysis too that we have with this project. So like from these single protein dynamics, like tracking their motion with live cell imaging, we can find organelle and cellular localization phenotypes, and then eventually from there we can kind of learn more about how peroxisomes maintain their pools in different aging contexts eventually for the biomedical side. And so thanks to everyone in my lab and my interfaces co-mentor, Elizabeth, and my department co-mentor, Vera, and then the the research tech at with the Rays, the Barboza lab, Julianne, helped with all my cell sorting for making the endogenous lines. So really couldn't have done that without her. Thank you.
Thank you, Dean, and thank you for bringing your mentor with you. Everyone's very welcome to invite their mentor and co-mentors and many members, and we're also free not to. But it's great to see them when they do come. So, questions for Dean?
I had a question. So you mentioned that, I guess, I might have mixed the two parts you talk up, but does PEX three also, might it also come for the mitochondria, or is there no evidence to suggest that?
Oh, did you mean like the PEC 16? Because that's
Yeah,
like,
oh,
okay,
right,
yeah.
PEX 16.
The PEC 16 has been proven like pretty definitively that it is going to the ER. Like there's been different like biochemistry studies that like show it's like being co-translationally inserted into the ER and everything. And I've never seen it anywhere but the ER, at least through imaging. But it's a curious question because if PEX 16 is recruiting PEX 3, then
Yeah,
how is PEX 3 getting into the mitochondria? Like, because I've definitely seen it in the mitochondria. So that's something that we're super interested in answering. And actually with the FRET assay, we're gonna be like looking at the actual like ratios and stuff of seeing that happen. And then we basically are going to like be doing this like high throughput like test of all like the interactors and stuff to see if you're like knocking out each of these individual proteins or like overexpressing them, if that changes like the ratios and making more mitochondrial localizations and stuff like that. Yeah. We think it might be something to do with like the hairpin structure of the protein, but we don't really know.
Interesting that it's still PEG 16 dependent.
Yeah,
Right?
yeah.
So PEG 3 insertion into mitochondria is also PEG 16 dependent, which when PEG 16 is at the ER.
does PEX three have a mitochondrial targeting sequence?
not.
No,
That's interesting.
it doesn't have ER targeting sequence either. Yeah.
So when you do this FRAP,
Hmm.
how much intensity you get, like if you look at it at the whole cell, if you want to do a flow cytometry, how is it possible to detect that is there a concentration limitation or
Sorry, for the FAP or for the flow cytometry?
because you said you want to apply this to a flow cytometry?
Oh, okay, yeah. No, so generally like when you're overexpressing the proteins, like they can like you can see it in the flow cytometry like fairly well, but basically like the assay would be like if it's tagged with if it's expressing one of the markers, because we have a green and a red, and so if it's expressing the green, it will be way out on this axis, and if it's expressing in the red, it will be out in this axis. And then if the fret is happening, it will basically like be like in a different section.
Is it cell cycle dependent? Like that's
The
live image.
Yeah, I guess the peroxisome biogenesis could be cell cycle dependent. That's not a specific phenomenon that I'm like looking at because necessarily, but it would make sense that when a cell is getting ready to divide, that there would need to be more peroxisomes made. I think the general assumption is that it's that like the growth and division model is just like accounts for that, but yeah, it's a good question.
It's my bias that couldn't help thinking how beautifully quantitative your data are and how great it would be for modeling, and
Yeah,
look forward to the day when I can see the data being analyzed by the Amara lab or the Rangamani lab.
Dr. Rangamani is like super excited for me to start getting the cryostructures and stuff, because I think it's definitely going to be very curvature dependent. All of these not like the protein localizations. I think that the way they're organizing is going to be super dependent on the ER curvature there. I think that that obviously the budding event is gonna be curvature dependent. Yeah, so I'm super excited like for all the collaboration with the interfaces.
So that's what the interfaces program is all about.
Yeah.
And both of you have given great examples of technology-driven, quantitative, multi-scale problems and beautiful presentations too. So thank you both very much. Thank you, Kelly.
Thank you.
I think we got. Can you talk about the side effects of the?
Yeah, everybody being so flexible with this. This was a big surprise, knew nothing about it and weren't sure until the very last minute that we could even stay in here. So I apologize for that, but thank you for understanding. And thank you for, like, Dr. McCulloch said, bringing a cheering section. It's always fun to